right, everybody, now we are going to move into looking at eukaryotic cells. Eukaryotic cells include plants, animals, protists, and fungi. Okay, and the first organelle that we're going to look at inside of a eukaryotic cell is the nucleus. An organelle just means that it is part of the cell, one of the functioning units that are inside of a cell. Okay, so we're looking at the one on the nucleus right now. The nucleus is located in all eukaryotic cells. So I think we will start right here. Okay, so as a reminder, we've got prokaryotic cells and we have eukaryotic cells. We've already looked at prokaryotic cells, which would be the bacteria and the archaea, and they do not have, first of all, they don't have any membrane-bound organelles, much less a nucleus, which is also a membrane-bound organelle. So what exactly is the job of the nucleus? Well, a lot of times when you're looking up the nucleus, it says that it's the boss, or it says that it's the control center, when in reality it's more like the library because of what it holds. It holds the DNA, which is the recipe or the instructions for the proteins that get made. Okay, so contains the DNA, and it is what determines what proteins need to be built when. Okay, the way that the DNA works is the first thing that happens is we keep the master copy. So we need to make a copy of it, and that's actually called RNA. So then the RNA will leave the nucleus through these what are called the pores. So if we look at this picture of the nucleus, let me make this a little bit smaller. We have all of these things in here called nuclear pores. I think I will color them, oh, it doesn't really matter, but I'm going to color a little bit. So I'm going to color these blue. So the nuclear pores are these like little holes that are inside of the nucleus. They kind of act like a tunnel, and parts of the RNA, now don't forget, I said the RNA is a copy of the DNA. The DNA is the master recipe, right? The main big dog, don't want to lose these instructions, right? So you kind of don't want to like mess them up, so we use them by first making a copy. And then that copy, that little piece of RNA, or the piece of RNA, actually leaves the nucleus through these things called the nuclear pores. Okay, and then where they head off to is something you already have heard me say is called the ribosomes. The ribosomes are like the little manufacturing units that actually build the proteins. So we have this. This is actually pointing to, so this whole thing is the nucleus, right? That's what this is trying to show. And this right here is known as the nucleolus. Okay, and the nucleolus is located inside the nucleus, along with the DNA. Okay, so that says nucleolus, and that's actually written for you right here. Okay, so in the nucleolus, what happens is these little pieces of RNA get built. I'm sorry, not RNA. The ribosomes get built. And the ribosomes are those, like I said, little pieces of machinery where the copy of the RNA gets read. RNA is that copy of the DNA. It gets read through here, and like this is like the little manufacturing plant right here. So we have both a large subunit and we have the small subunit. So you have the DNA inside of the nucleus, and the DNA is going to decide what proteins need to be built. The first thing that happens is this copy of it gets made, and it's going to leave through the nuclear pores, and it's going to seek out these little ribosomes, these little ribosome manufacturing plants. Now, the ribosome manufacturing plants, these pieces also get made inside the nucleus, but specifically inside the nucleolus. Okay, so this is the spot, or the job of the nucleolus is the area where the ribosomes get built. So the DNA, which is inside the nucleus, right, a copy of it gets made. That's called RNA. We're going to learn a lot about that more later. And it's going to leave the nucleus, right? And so how does it leave the nucleus? Well, it leaves through these little channels or these openings that kind of act like tunnels from the inside of the nucleus to the outside of the nucleus. The thing is, the nucleus, along with a lot of the different components of eukaryotic cells, are membrane-bound organelles, which means they all have their own membrane. Well, why is something like that important? Why would it be important for the nucleus to be membrane-bound? Well, every time you have an extra level of anything that separates the inside from the outside of something, so here it would be the inside of the nucleus where the DNA is, where the nucleolus is, and the outside of it is the rest of the cell. Anytime you have this membrane-bound organelle, when you have a membrane, it just offers extra protection. How is it, or rather what exactly is this membrane, right? So here it is called the nuclear envelope. And we've already learned about macromolecules, and one of the things that we learned about macromolecules is that there's something called carbohydrates, and then there was something called lipids. This nuclear envelope is made up of what is called a double layer of membrane, where we have these things that are being drawn like this. And then we've got, like, here's the top of some, and then there's more, right? So that's what these are. This is a carbohydrate head and the lipid tail. Okay, so this is a carb head, this is made of carbs, and this is made of lipids. Okay, and the same with down here. So all of these are the carbs, and then the inside are the lipids. Well, the thing is, the lipid part, the lipid tails, they are hydrophilic, meaning I'm sorry, I got that backwards. They are hydrophobic, meaning that the inside right here does not like water. The lipid part, right, because we kind of learned oils and waters don't mix, right? They are hydrophobic. They don't mix well with water. However, the carb portion, the head part, actually likes water. So this idea of having part likes water, part hate water, and a double membrane, all of that together works to keep things in and outside of the nucleus. The thing that's really interesting about the DNA is that in one cell, I just looked this up, in one cell, or the nucleus, just one, and we have trillions of cells. In just one cell is somewhere between 2 and 3 meters of DNA. 2 meters is about 6 feet. 3 meters is about 9 feet. So if we took and untangled our DNA and we were able to lay it out in one straight line, we would have somewhere between 6 and 9 feet. And that is just in one single cell. It is crazy. So how does it fit? Like, we're not even talking about the cell, really. It fits inside of the nucleus, right? So it's not even the entire size of a cell, because the nucleus is part of the cell, so the cell itself is bigger than the nucleus, and we're talking about all of this between 6 and 9 feet of thread-like material fits inside of the nucleus. Well, it just folds and folds and folds and folds and folds and folds on top of itself until it takes hardly any space at all. Okay, so really the only little bit extra to put is this over here. So this is where, this right here is an example of the RNA. Go like that. Okay, so it's the RNA. This is the copy of the DNA that is able to leave through the nuclear pore. Okay, and then here's another example of one right here. So RNA is the copy. There's actually more than one type, so I'm just leaving it real general right now. As you can see, it's called mRNA, messenger RNA, but for now I'm just going to call it RNA. We're going to be learning more about that later.